Record. There we go. We missed the whole introduction there. So welcome, everybody. I'm passing this over to Jim Prince to introduce our guest speaker tonight.
Yeah, good evening, folks. Lovely spring. Feels like summer evening tonight. As Deb said, we're going to talk about bugs in our muck with Sadie Fischer. Welcome. We do benthic biomonitoring on the Kennisis lakes, as do a number of other lakes in the Haliburton region with the help of lake associations. I think we're up to 25 or so. And Sadie will tell you about why we look at the bugs in our muck and what story that tells about our lake health. About our webinar series, we started the webinar series last year. We did a webinar on on algal blooms, and we actually shifted in the fall and did a webinar with the candidates for Ward 4, which worked out very well. But we have some other proposed topics, and you know, after tonight, please do send in your comments. We're looking at boating safety, pesticides in our shoreline, renting your cottage, new to the Kennisis Lakes. Have a look at those. And if you've got other suggestions for webinars, please do send them in. We have a webinar committee, thanks to those folks, and we decide what we're going to present. And usually we try one in the spring and one in the fall. So tonight's agenda, we're in the welcome portion now. And what's up, Kennisis backgrounder? We do have a kids contest tonight, which is a little bit of a departure from our normal webinars. And I'm going to provide the details of that next. And then Sadie will be up as our guest speaker talking about the benthic biomonitoring. And then what we'll do is we'll have questions from participants. So when you get to that point, you can unmute, turn your camera on and ask the questions. And if you feel motivated, you can also put questions in during chat. We'll try to cover those off as well. I'm going to try and be out of here by 8 o'clock. And the motivation for doing this particular topic comes from our 2021 survey, which showed that 99% of respondents say maintaining our excellent water quality is very important. And one measure of that is the benthic health on our lakes. So about the contest. So the kids that are on, and I hope there's a few out there, and some of you kids, if you feel like acting like kids, you can do this as well, some of you older folks. We're actually going to ask you to pick one of the bugs in our muck, or a couple of them, that Sadie will talk about and you might find interesting, and send in some hand-drawn artwork or any media you would like, any medium you would like, and include the name of your critters somewhere on the artwork, on the front or the back. When your drawing, sketch, or artwork is complete, have your folks submit it in an image of the artwork, including the artist's name, to the KLCOA at klcoainfo@gmail.com. If you have trouble with that, use Deb's email or my email, but shoot for that one. A panel of judges, including Sadie, will select the 3 top submissions, and selected artists will win a special prize that can be redeemed at Kennisis Marina. There's a hint there about what it might be. And please submit your artwork by Wednesday, June the 14th. The reason for that is we want to actually do the judging and get the certificates back out to winners announced by July the 1st. So if you have any questions, you can bring those up at the end as well. So I'm going to turn it over to Sadie to talk about the benthic biomonitoring. By way of introduction, Sadie's the environmental program coordinator with Woodlands and Waterways EcoWatch, which is a part of ULINK's Community Center for Research. Sadie has been in steering the ship at ULINKS. I've worked with ULINKS, I'm on their board, for community-based research for a whole variety of topics, including socioeconomic, health and safety, and particularly environmental projects like the benthic biomonitoring, water quality, which we participate in on the Kennisis Lakes, and some terrestrial monitoring in forest plots in the Haliburton Highlands plots. And there's one really cool one we're looking at right now, or Sadie's looking at right now, is developing a seed bank for native plants on Nora's Islands. That's very close to home for us. So off you go, Sadie. Have fun.
Awesome. Thanks so much, Jim, for the introduction. It's a pleasure to be here and talk about this. It's something I've been doing for the last 3 years since I've been at ULINKS. I've absolutely loved it. So I'm excited to share it all with you. And throughout the slides, you will see some pretty good examples of photos of different critters for some inspiration for your drawings. So we'll get started there, Jim. Next slide. Just a little bit about what I'm going to talk about today. I'll do a little introduction about myself and what makes me qualified to talk about this with you guys. A brief introduction to our woodlands and waterways programming. Then I'll get into what are macroinvertebrates, why we monitor them, how we monitor them, and what they are telling us about Kennisis Lake and the bigger picture in Haliburton Catty. Next slide. So I do have some educational background in this sort of work. I went to Lakehead University, where I got my Honors Bachelor of Environmental Earth Sciences. I also decided to go to Fleming College because I wanted a little bit more hands-on work. So I got my Ecosystem Management Tech diploma from there. Through school and as well through different work experiences, I've obtained quite a few different certifications. So the first one you see there is OBBN. That one relates directly to our benthic macroinvertebrates. And then I also have some other things here, such as electrofishing, invasive species management. I've done some habitat restoration. And I've done all of this with different nonprofit conservation authorities or research organizations, anywhere from downtown Toronto up to northern Manitoba at the research center there. So I like to think I have quite a bit of experience in Canadian research, which has been truly amazing. However, the last 3 years I've been with ULinks and doing a lot of the programming that Jim had mentioned. On a more personal note, love being outside, hiking, camping. I'm trying to teach myself a whole bunch of new skills lately. Most recently has been stained glass and rollerblading. I don't know how many adults have tried to teach themselves rollerblading lately, but it is not as easy as I remember it. So that's been a lot of fun. Next slide. So I'm blocking everything here. So in 2019, ULinks was getting a lot of questions from the community, and they were all the same questions from mostly lake associations, is how can we monitor the health of our lakes to ensure the water quality? Exactly what you guys were wondering in your survey. 99% of you are on the same page. So how can we collect and store that data for the region? So what we did was we established the Woodlands and Waterways EcoWatch. Now this is a program that hosts a bunch of different research, such as benthic biomonitoring. And we try to involve as many community members as possible. So what we've done is we've developed protocols, established sites on your lakes and forests, and we bring in community members and students from our partner institutions, such as Trent and Fleming, to help do the monitoring on the lake. And lastly, we want to ensure that data is used for decision making. So we're giving all of that information we're collecting right back to the Lake Association, whether it's in a report or a poster. So you guys can take that data and be like, are what we're doing correct? Can we change some of our habits to ensure the health of these lakes? Next slide. So these are just some of the examples of things we do. The top left there is a couple of students from Trent doing some benthic biomonitoring on KLCOA Lake, not too far from you guys. We also take students into the forest to our different land trust properties to measure and monitor the health of our forests and the trees, to see if there's any effects due to climate change or insects, or if they're flourishing long term. So we go back to those properties every couple of years to make sure things are good. In the bottom there, we had a student out at one of our wetlands looking for species at risk, taking some photos. That was a really fun day. And the last photo there is actually one of the workshops that we host for our community partners and anyone who wants to be engaged. This was a training session for benthic sampling. We also hosted a second one where some of our community volunteers got certified with OBBN, and we hope to do that again one day. Next slide. Okay, so what are benthic macroinvertebrates? So what I've done here is actually broken down that word for you. Benthic means bottom dwelling, so anything that's living on the bottom of the lake. That could be at the very bottom of the depths, or more likely along the shorelines where there's some light penetration. Macro, so you can see with the naked eye. that doesn't mean it's going to be huge. So we have two examples here for you. We have our crayfish, which I'm sure many of you are very familiar with. Can get pretty large, hold them in the palm of your hand. But we also have one of our smaller organisms, the mite, on the left-hand side. Now that's on someone's tip of their finger. You can actually see their fingerprint, and it's encased in a water droplet. Teeny, teeny, tiny. You can see it with your eye, but sometimes you need a little bit of help. And lastly, invertebrates, so any organisms without backbones. So the crayfish is a crustacean, and they have that outer harder shell. Some of them don't even have that. They're just more soft and fleshy. So what or how are these benthics important to your lake ecosystem? So they're actually really important when it comes to the food web within the waters. So at the bottom here, you see your phytoplanktons, your organic materials. That's what these invertebrates eat. Many of them eat. So they take it from that plant life into animal life. So some of the examples we have here are stoneflies or dragonflies and midges. So these invertebrates, they will eat the plants or each other, and then they'll be eaten by the fish. So very, very important in the overall food web. Next slide. And what is biomonitoring? That's another big word that we have that we're going to be saying a lot today. Essentially, it's just biological monitoring. So instead of collecting chemistry measurements or physical measurements, we're actually looking at living organisms. And the main way we do that is we're looking at how they respond to certain pollutants that might be going into the water, or disruption to the actual habitat. Maybe a shoreline's being cleared for a new dock. That can really affect the different communities and species that live there. So we take a look at what we have and what we don't have, and the presence or absence of certain species really gives us an idea of how healthy that specific site is. And when we're talking about biomonitoring in the bigger picture and looking at long-term monitoring, taking those measurements for several years, it's like going to a doctor's for a regular checkup. Going in every couple of years, like most of us should be doing. Might not be doing it all the time. Going in, they're taking our blood pressure, checking to see how our reflexes are, hitting our knee. It's the same thing that we're doing, just on these small critters. Next slide. So why is it the benthics that we focus on when it comes to biomonitoring? There's a couple really great reasons for that. And it's because one of the first ones is they live, if not all of their life, but a large portion of their life within the water. So a really good example of that is the dragonfly that you see on the left-hand side. So their eggs are in the water, their larva state is in the water, and they can be in the water for up to 2 years or so. They crawl out, they molt, turn into adults that you see all the time around the lake, they mate, and then right back into the water at a new cycle. So because they're in the water all the time, or majority of their time, they're really being affected by other inputs in that water. There's a really great example of a large specimen of a dragonfly larva at the bottom there. So that's a really good example of macro. They can get big, but I've also seen dragonflies really, really tiny as well. So also when we're looking for something to monitor, we want to know how many different groups or families or species of that level, I guess. So for OBBN, which is Ontario Benthos Biomonitoring Network, that's the protocol we follow. There's 27 different groups that we focus in on. Now those can be broken up in all these different families, and the list goes on and on, breaking them up. But for OBBN purposes, we keep it to that 27 group level, so it's easier to identify, and it gives us all the information we need when it comes to understanding what those groups mean when it comes to the lake health. And they're also they're found everywhere in water. They're in our streams, they're in our lakes, they're in our wetlands. So no matter what type of ecosystem you want to focus on, you'll be able to find benthics. And a really important point here is that they're in large numbers. We don't want to, when we're sampling and analyzing any type of animal, remove them from their environment if there's going to be limited amounts of them. So there's thousands, if not millions, of these little guys in your lake. So taking a little sample every year is not going to harm the population. And lastly, they don't move around too much, where fish, you'll find, will go all around the lake. These little guys are pretty sedentary. They'll stay in their general location. So if there is pollution close by, we'll be able to pinpoint that a little bit easier. Next slide. So these are just some of the protocols that we follow. So OBBN protocol and how we monitor them. So when it comes to protocols, they're developed so that we are sampling the exact same way, so that everything that we collect, all that information is actually comparable. Because if we have one person sampling one way, someone else sampling a different way, they're not going to be representative of each other, and we can't compare them. So we follow a very strict protocol that was developed in about 2005, I believe, by Chris Jones at Dorset. And you can modify them a little bit, but as long as you have the okay from them, this is what we do. So the first thing is we don't even get in the water yet. We want to take a really good look at the site features, things going on in the shoreline around the water. So you'll actually see me pointing there to different vegetation. We can have wetland species, trees, meadow environment, scrubland, or nothing at all. You can actually see the water levels are low at that site, so we're standing in muck. And we're also collecting habitat features in the water too, so substrates, so whether it's sand, gravel, bedrock, that sort of thing. If there's aquatic vegetation for the critters to eat. And looking at the size of those pebbles and algae. In the next photo, you'll actually see us getting into the water. So what we like to do is measure the distance that we actually collect these bugs from. The distance from shoreline doesn't matter. It's how deep you want to get. Because as I mentioned earlier, all of these bugs like to be along that shoreline because they get the penetration from the sun, where it's warmer, where there's more vegetation for them to eat. So we're measuring to see how far that is. In the next photo, you'll see one of our community volunteers actually doing collection. So we have these nets, they're specific size, so none of the little guys go through the net, but we can collect all the bigger specimen as well. And what she's doing is picking up the substrate, kicking up with her feet, and then scooping up with the net everything that gets kicked into the air. In the next photo here, you'll see us taking that entire sample and putting it in a jar. So there's two things that you can do here is you can put them in the jar, you can look and try and ID all these species on site, so you can throw it back into the water, or you can preserve it and take it into the lab with you later. If it's a nice sunny day, I suggest doing it on the shoreline, but most of the time we do take it to the lab so we can use our microscope to help us analyze it. The next photo, you'll see us doing a quick double check there. So you'd never want to leave your site if you don't have enough of the sample to actually analyze. So what I like to do is I like to take our sample, take a really good look at it, and see how much movement you have going on. Because you'll see the little guys trying to climb out of the substrate there. You also want to check for any fish, the invasive species. We return all fish, frogs, tadpoles back into the water. And then also, like I mentioned, you want to make sure you have enough to look at. You can usually see about 10% of what you actually have. So if I can count 10 bugs moving around, then we're good to go. If I can only see 1 or 2, we do everything all over again. We go back into the water, measure it out, take in sweep, collect it again, and put it back in the jar. Next slide. So this photo was actually taken from our last certification course. That's Barb Elliott, if any of you know her, who was the instructor. So after we collect our samples, we take them inside. And what we do is we take 100 organisms out of each sample, look at them at the microscope, identify them, put them in a tally, and we save those numbers for later. We also do collect all of the samples and keep them in a library at our office in case we want to refer to them later or have maybe a much larger research project on how many? Over 300 samples we probably have now. Next slide. And the last thing we do once we have all of that collected is we need to do some quality assurance and make sure that what we're saying we actually have. So anyone with OBBN certification will take the samples that we've had collected, whether it be by a student or a volunteer or even staff, and then we take a second look at it, confirm the ID, and then write that down, use that for analysis. And we also have to have an OBBN certified person do that so that we can upload it into the Ontario database where all of this information is kept. Next slide. So that brings us to Kennisis Lake and what we've done here. So as you can see, we have 6 different sites on your lake. They're not really in order, so don't mind remembering. But we like to have sites across the entire lake. This will give us a much bigger representation of what's going on throughout the entire thing. Because Kennisis is so large, we needed at least 6 of them to make sure that we're covering all of the areas. And depending on what your research question is, if you have a site and you know that you're going to have some big long-term construction going on there, what you can even do is take some samples before construction starts, maybe during construction, and then a couple of years down the road to see how that disturbances have affected the habitat and the communities that you have in the water, which is really interesting. Next slide. Okay, so at Kennisis Lake, we've done 4 years of sampling there. Because Kennisis was one of the first lakes that we've done the programming on, we used 2019 more as a pilot season. So that was to ensure that we were doing all of the protocols properly, that we're getting enough information from the sites themselves. So we had 2 sites there, one at Paddy's Bay and one from Johnson Lake, which is more of a reference site, something that we compare Paddy's Bay to. And then after 2019, we did a full set of sampling sessions. So that's all 6 sites that I showed you in the last slide. And from that, we've collected over 3,600 organisms for analysis. You'll see on the right-hand side the 2 different types of bugs you got there. Those are the 2 that are probably most popular within Kennisis Lake. And if not just Kennisis Lake, I'd say all lakes in Haliburton, or maybe all lakes in general. On the top, you have your amphipods, which are your scuds. They're going to be everywhere. They love silts. They love sands on sandy beaches. They love warmer waters, which lakes tend to be compared to streams. So they are very happy. And sometimes we collect a sample and they can be up to 75% of those little guys. And underneath there, you have our non-biting midges, or ornaments. They're really happy in wetlands, mucky substrates, very similar to the scuds. And they're really interesting because you'll take a look at them, and this guy's upside down, but they have that very prominent J shape, so they're really easy to identify underneath the microscope. Now, when you have, we'll just go back for 1 second. When you have all this information that you're collecting, what you can do with it is you can do either spatial or temporal analysis. So spatial is analyzing from site to site over space, and then temporal is over time. So seeing how one site changes from year to year long term. We're currently doing both, but we do recommend to have at least 5 years of data for proper temporal analysis. That way we can really understand those natural variances that occur from year to year. One year you might have drought, the next year you might have really heavy rainfall or cooler temperatures. So we want to account for all of that variability beforehand before we can really say, this is the normal for this lake. Next slide. Okay, so once we collect all that information, we record what we have, which groups they're in, we put some of those numbers into formulas to interpret the water and ecosystem health. So I've listed 2 different indices that we use. The first one we have is percent EOT. So EOT stands for our top 3 guys here that I like to refer to a lot. So that's our Ephemeroptera for E, which is our mayflies, our Odonata, which is our damselflies and dragonflies. And lastly, our Trichoptera on the end there is the caddisfly. So I think those two got switched on the slide, but that's okay. So the last one, the casing with all of those little pebbles, that's our caddisfly. And these three groupings, they are much more sensitive to inputs, whether it's organic or inorganic pollutants, those disturbances, those physical disturbances on the shorelines. So essentially, the more of these that we see, it interprets a healthier ecosystem. So we want those percentages to be up. So that's the first indice we look at. The second one refers to diversity. So that's Simpson's Diversity Index. It measures the diversity of the different taxa, but that takes into account abundance and richness. So how many, or all the different types of groups that we have, and how many of each individual is within those groups. So again, higher the diversity, usually the more, the healthier the habitat and the ecosystem. Next slide. So I have a couple results for you guys from Kennisis Lake. So these are the different percent EOT results we had since 2022. So first, when you look at this graph, we have the different colors in the background. So what we've done actually is taken results from Halliburton County lakes, as well as those within the region, Muskoka area, Kawarthas, and we've taken a random sample and determined a normal range for the county. So that normal range is about 4.5 to about 37%. And that's what we typically find. Now the yellow represents atypical, which is some areas that we might be a little bit more concerned about, especially if it's on the lower side. And then our extremely atypical, which is this is really out of the norm. They should be much higher. Now, on the other side of things, we have the atypical and extremely atypical on the top levels as well. So over that 37%, I think to about 52%, and then 52% and higher. Those aren't necessarily bad results if we fall in those categories. It's just unusual. It's higher than we'd expect. So it's something we want to keep our eye on and maybe figure out why later on. As you can see here, all of those levels fell well within the typical range. We do see a pretty significant increase. But as I mentioned before, we want about 2 years more of information to really put a strong trend line and say that the percentage of those species are increasing. Next slide. So secondly, here we have the Simpson's diversity of Kennisis Lake for the same 3 years. So how we read this is 0 is no diversity and 1 is infinite diversity. So anything below 0.4, I would say is in a concerned area. From 0.4 to 0.6 is the fair to good area. And then anything greater than 0.6 is pretty great. That's where we like to see things. And as you can see, Kennisis Lake is in a pretty great spot at the moment. Interesting enough, you have that same increase from 2020 to 2021 with a slight drop in 2022. Again, it's still in that very good range, so I wouldn't have any concern at this point. Next slide. So this one is the spatial comparison, so from site to site for 2022. I tried to combine both of those results on one chart, so I apologize if it's a little messy or hard to read, but I'll walk you through it. So Simpson's diversity is on the left-hand column, and that's all of the bar graphs, so all of the blue ones. Again, you're looking straight across. Everything is above 0.6, close to 0.7 and higher. So diversity across your lake is looking really good. There's no one site that we monitor that needs to be watched a little bit closer at this point. However, you look at Kennisis 7, that site there, and it's pretty high. It's above 0.8, so there must be something good going on there. And then you look at percent EOT, and that's on the the right-hand column or the right-hand axis. If we remember, we want to be above about 4.5 5 or so, and we're all the way already above 10% at the lowest on Kennisis site 2. So no area for concern there. And then, as I mentioned earlier, that upper atypical area, we have 3 sites there. So Kennisis 4, 7, and 8. So that's really interesting to see. Sometimes you'll see it maybe at one site where maybe some eggs had just hatched and there's a larger community of them. But seeing it across there is really interesting, and we should keep an eye on those for next year to see how they compare and if it's very similar throughout. Yeah, but it's interesting to see how some of the higher levels of diversity have the lower levels of percent EOT, like at Kennisis 1 and 2. So that's really important to use different indices. We don't want to look at just one thing and take that for certain. Having several ones to look at and analyze is really good at, gives us a better picture overall. Next slide. So bigger picture. So what are we doing for Halliburton region, including Kennisis Lake? So this is a really good comparison. And prior to 2019, when this image was taken off on the left, that's actually from the OBVN database itself. So each of those little orange dots are sites on different lakes. So you'll see that big clump in the top corner, that's your Muskoka area. They have the Muskoka Watershed Council. So they're doing a lot of work there. If you look lower to the bottom, we have the Kawartha region, the Kawartha Conservation Authority, or even the Simcoe, Lake Simcoe Conservation Authority. They do all of this sampling as well. So they have a whole bunch of dots. As some of you know, there is no Conservation Authority or Watershed Council in Halliburton. So that's why we decided to get the ball rolling there until there is. So on the right-hand photo, you'll see the 25 lakes that we sample currently. And those aren't even the sites. There's at least 3 to 7 sites per lake, so up to 81 sites. So when that database gets updated in the next couple of years, we're going to see a whole bunch more dots on there. Next slide. So what's next for you guys on Kennisis Lake? So we're going to continue collecting for 5 more years to develop that baseline inventory and figure out what really is normal for your lake. And depending on the normal results or what kind of trends we see in that 5-year period, that'll determine how often we need to come back and continue sampling. Ideally, we want to see a nice straight consistency across the board. Things are healthy, things are good, or even a increasing trajectory, things are getting even better. And if that's the case, then we might do some of the sampling, maybe every 5, every 10 years, depending on what's going on the lake. However, if we're seeing some more of those negative trends, things are getting a little bit unhealthier, we're not sure why, we might continue to do this every 2-3 years to really keep understanding the trajectory of those trends. We also want to compare the lakes across Halliburton County to really see how they are compared to each other, because we're living in watersheds. All of our lakes and rivers and streams are connected. So if one polluted area is draining into another, we want to be able to figure that out. At this point, we don't have those exact results for you guys, but in the next year or 2 we will, and we'll be able to really compare some of our pilot lakes across the watershed. Next slide. You guys want to get involved? We're always looking for volunteers. We got a couple photographed there, 2 guys from the Canadian Conservation Corps. They came out to Kennisis Lake last year and helped us do some sampling. We do it every September, or between August and Thanksgiving weekend, where we collect samples. If you guys want to come out and meet us at some of our sites, we'd be happy to do it. I always have to throw this in there too. We always accept donations. I know the Lake Association donates to us every year, but we always accept those. So thank you so much. Okay, does anyone have any questions?
Thanks, Sadie. We do have two questions that have come in via chat, so I'll read those, and anybody that wants to unmute after this can go ahead and actually ask your questions. So the first one is from Chris. Why no sample sites on Little Kennisis?
I don't know, Chris. Unfortunately, I did not set up the sites on these lakes. However, I don't see an issue in adding one on Kennisis Lake. I don't think we need several because of the size of it. But you're right. I think it's important to have that there to ensure, to make sure we're covering all of our bases on the lakes themselves. So that's something we could set up this year. I would suggest somewhere that is accessible by land. It doesn't have to be private or sorry, public. If someone wants to volunteer their shoreline to us, we can come out, and it takes about an hour to do sampling there, and you guys can join the event as well. Jim, maybe I'll have you suggest an area.
There was a reason. In 2019, when we started, we actually had more sites than we currently sample. And we actually had a little bit of trouble coordinating boats to get to the non-road access sites. When we looked at Little K, there were only two real places on Little K that was public land where we didn't have to actually coordinate with somebody. That was at the outlet of Wolf Lake and the small patch of Watts Road, which is right beside the lake on the first road into Watts. So yes, absolutely, we could throw a couple Little K in there. But when we first started, we did prioritize areas that we could get to from the road, and there were lots of those on Big K. So that was the reason.
Yeah, in the past, we had several sites on lakes, and the last couple of years with COVID, people taking their boats out of the waters at different times, we figure it's best to do as many land access sites as possible. That way we're not harassing you to taxi us around.
Okay, and we have one other question that came in via chat from Julie. Do invertebrates go through population cycles?
Yes, I believe they do. You mean by having larger populations certain times and then they die off after a couple of years? So it depends on the species. Certain ones their cycle lasts 1 year, others a couple years. I think it's crayfish up to 5 to 6 years. So it does vary between species. And I think looking at the big three that we like to focus on, the mayflies are 1 to 2 years, caddisflies are similar, and the damselflies and dragonflies are about the same. Yeah.
Sorry, Sadie.
Variation.
Oh, it's Julie. Can I clarify my question?
Of course.
I guess I was asking, are their populations susceptible to cycles, like because of temperature or the ice going out earlier? Are they susceptible? So some years their populations are lower or higher other times? That's what I guess I was asking with not very clear words.
Yeah, I feel like they are, because they last several years in the water. They do go through the on ice periods. They kind of hibernate a little bit. They do their thing. They're much slower in the waters, but they're much more affected by the temperature and water levels we're finding. If you're having a lot less rainfall and the lake levels are lowering, which we see a lot in our reservoir lakes, then that's definitely going to affect the amount of species we find in that small little benthic zone that we look at. And I saw in the chat there that the crayfish are making a comeback. So I thought that was related to that question, but no, Julie, we do see that sometimes. So climate and weather patterns are huge in looking at this at the bigger picture. So yes.
So can I add something to that? Then would lakes like ours that draw like that, the Trent Severn draws a lot, like almost 6 feet, would that do you take that into account when you're comparing our lake to other lakes that may not have that draw?
Yes, so we definitely do. We don't have the specific results for you yet, but our plans with all of our lakes right now is to do more statistical analysis with the results, and that does incorporate the drawdown lake versus the non-drawdown lakes. I would like to say yes, there is an effect. I don't see how there couldn't be, because that water is drawing down faster than some of these critters can move. So it'll be really interesting. I think after next year, a lot of our lakes from that 5-year period. So we'll have those answers for you.
Great, thank you. Thanks.
Okay, and there's another question here from Chris. How do the different bugs help the lakes?
So how they help lakes, I think the main thing for the different types of bug is just the food cycle. So certain bugs, they convert that plant matter that they eat, whether it's the leaves that are broken down or the aquatic vegetation, and they turn that into energy, which goes up into the fish, into the ducks, that sort of thing. And then they provide food for each other. They decompose back into the water. Trying to think. So there's some like the bivalves, so like our clams, our mussels, they can help clean water and make it clearer. We also have bugs that don't help so much, such as our invasive species. So that was like our zebra mussels, our mystery snails, we see those too, and we try to limit those from spreading around. So that's the other side of that question.
And also now the crayfish question. Crayfish seem to be making a comeback. What does that indicate?
So from my research, I don't really use crayfish specifically at indicators for lake health. However, because they have that much harder shell on them, which uses, oh, no, I'm confusing that. So I apologize. They are one of our larger species who prey on a lot of the smaller species. So seeing those crayfish means that there's probably more food for them in the environments. So a lot of our smaller scuds or midges, some of that stuff. So it might just be an overall healthier boom at this point, and it could just be that cycle that we're seeing of more prey, more predators, etc. But I haven't actually seen that from my end of things for crayfish, but I'm excited to see what the results are in the fall to see how many more that we catch.
It's Jim here. There's also one other competing issue in our lakes is we have a fairly significant calcium decline. And that calcium decline has been measured and it's evident. And the fact that we've got less calcium doesn't fare well for the crayfish, which use calcium to build their exoskeleton. So that's a competing issue with crayfish as well.
Yeah, so if we're seeing more crayfish, then that indicates that there might be more calcium available. Yeah. That's really interesting. And that's something that you could test if you'd like, if you wanted to measure the calcium in the water by taking some water chemistry samples and comparing that, that could be really interesting.
Does anybody else have questions? We don't have any more in the chat.
Yeah, if anybody does, just unmute and fire away. I did have one question directly from Theo, who's watching tonight, and he asked, what age is the contest up to? And I said, well, it doesn't really matter too much. How old you are, you could probably submit, but it's mostly for kids.
I know I'm going to submit one just for fun.
I think I'm going to submit one too. Okay, if no more questions, we can move on. So this again, a reminder, I don't think we've got too many kids on tonight, but we'll probably push this out in an e-blast for people to watch the video and have a look and submit drawings. So please do send in your drawings, bugs in our muck, submit them to klcoinfo@gmail.com by June 14th. And again, we'll select the winners by July 1st. And thank you very much for attending. So a couple of final announcements. So this is Sadie's parting shot. Sadie is actually leaving ULinks. Her last day is Friday, I believe. And this is one of her last tasks at that job. She's moved to Saskatoon to start a new career and a new journey in a new job. So thanks again, Sadie, for that. Great working with you. Finally, thanks to the KLCOA board and volunteers for all the support with their lake health initiatives. There's a couple people on the call tonight who are deeply involved in things like water quality testing and other programs. So great, good stuff. And Sadie, thanks to you for presenting tonight. That was actually really great. Good balance, especially for kids that are watching. It was really nice. And thanks to all of you for attending. Keep an eye out for the bugs in our muck. And kids, send in that artwork and have a great evening, everybody.
Thanks so much, everyone, for having me.
Thank you, everyone, for joining us tonight.